in biological research across world. How does the cell make specific RNAs at specific time points and at specific places in an organism? Just to give you another example, this is a picture of a mice skeleton. You can see the 5 fingers of a mice hand, an arm of a mice. When this mice was an embryo, a particular gene, I'm not going to tell you the name of the gene, but that gene is expressed in this bulge, which later develops into a hand, right? So this gene functions in making this skeleton or making this anatomy. When we modify that expression pattern, as you see here in the bottom figure, using techniques and cause this gene to express in areas outside the bulb, you can see that the gene, this change causes an extra finger in the hand of the mice. So when we change expression pattern and make it broader, mice develops an extra finger. Keep in mind that this gene is not changed. The gene and the RNA that is made is still the same. It is only the area or where in the developing embryo that gene is expressed or that RNA is made. Similarly, you can think of all sorts of problems that can occur if RNA is not made correctly or RNA is made at a wrong location in the organism. And these kind of events happen, it leads to disease. And this particular case of extra finger is not really a disease. It doesn't cause anything else, but you can think of all sorts of disabilities or all sorts of intellectual disorders that can happen if RNAs are not expressed at the right place at the right time. So it is crucial for us to understand how RNAs are made in our cells, for us to understand diseases better, and for us to make therapies for diseases. With that said, I can now go into my research. Like I said before, our transcription is the process of making RNA from DNA. You can see a picture, a schematic representation of this here. A protein called RNA polymerase makes RNA from DNA. Just like when we write something or when we copy something from a reference book, sometimes mistakes are made in RNA synthesis. So this is what I'm trying to show here in the bottom schematic. You can see a couple of mistakes on this RNA strand that is made now. Our cells have a system in place to identify these RNAs with mistake. So when an RNA is made and it has a mistake, our cells are able to identify that and remove that so that there are no adverse effects like the one I showed you before. So this process is very much like a spell check on Microsoft Word. Let's say you type in a word with a wrong spelling, the word can automatically detect that it's wrong and correct it, right? So our cells have an automatic spell check system to make sure that we make the correct RNA and not the wrong RNA. So this process in cells is called nonsense-mediated mRNA decay. I don't want you guys to probably remember the name, but just to put it out there, it is a process called nonsense-mediated decay, or NMD in short. So how do I do this? How do we do this in the lab? So what we actually do is grow human cells in culture. So this is what I've shown here on the top right. Human cells are grown in petri dishes in very sterile and very protected conditions inside the lab. This is a fume hood where we do this. And these cells are grown in incubators shown here with specific carbon dioxide and oxygen levels and temperatures maintained. What I do is I make changes in the DNA of these cells to figure out how RNA of those cells change. So I make changes in the DNA of these cells and then isolate RNA from these modified cells and try to analyze how different RNA levels change. And that is how I understand how proteins in the nonsense-mediated decay pathway works. So for this, we multiple approaches in the lab. This is a picture of my lab on the top right. we take out RNA and do all sorts of biochemical experiments, most of which is complex and I cannot explain to you guys over a short period of time. But understand that we have techniques now in our lab, in our world, to take a cell and break it open and then look at what RNAs are inside a cell. So we do all that to figure out how different proteins play a role in this spell check system. We also use computational approaches um, to study this. Another thing that I'm doing in the lab is study how cells handle the RNA. RNA, as I showed before, uh, is usually shown in high school textbooks or undergrad textbook as a ribbon-like structure that is always straight or wiggly. Um, but there are like thousands of genes in our cells. There are thousands of RNAs that are made in our cells, and if they are all straight like that, they can all intertangle with each other and create a problem for the cell. So if you look at So because of this problem, RNA doesn't exist in cells as single ribbon-like structures. If you look at electron microscope images of RNA, they are never ribbon-like structure as shown here. They are actually globular and kind of rod-like structures inside the cell. So RNAs inside cells are packed into RNA protein complexes like a schematic shown here, and we currently do not understand how this is achieved, how cells are able to make an RNA and make it into a rod, pack proteins into it and make a rod like structure. We don't know that, and I try to study this process also in cells, and I use similar techniques. So this is so we are running short of time, but this is mostly what I will be telling, what I have for you in terms of research. Now I wanted to tell you about why doing all this is important. So it is easily understood that in the current situation of COVID, right, we need more biology research lives. Do you guys know what the first vaccine of COVID was in the US? It is actually an mRNA vaccine called from a company called Moderna and Pfizer. These vaccines are actual RNA molecules that we inject into our muscle, and ribosomes inside cells, like proteins inside our cell, using this mRNA, they make proteins that are specific to the COVID virus. And our body recognizes these proteins and remembers them so that when we actually are infected with the actual virus, we have a better immune system against it. So these companies were able to make this vaccine within 3 months after COVID genome was published. They were